Again, a very good evening to all of you. I hope all of you are doing well at your places. Today, I'm going to talk about ion torrent sequencing. Ion torrent sequencing is a second generation sequencing technique. Last time, I had talked about nanopore, which is the third generation sequencing technique. But remember, we had left with a second generation sequencing technique, and that is the ion torrent sequencing technique. So Ion Torrent sequencing technique, there are some quick facts, or this slide is showing you something about some quick facts about the Ion Torrent sequencing. So one of the quick facts is that the Ion Torrent sequencing is similar to the other second generation sequencing techniques, that is the 454 sequencing or the Illumina sequencing. There is a spelling error. It is not Illumini, it is the Illumina sequencing. So here is Illumina, not Illumini. So it is similar to the 454 and Illumina sequencing in one of the aspect. And what is that aspect? That there is sequence by synthesis. हमने क्या देखा था इन नैनोपोर सीक्वेंसिंग वी हैड सीन दैट वहां पर सिंथेसिस नहीं होती है ओके the molecules or the nucleotides they move into the nanopore, they move through the nanopore, and the second strand synthesis does not take place. However, in case of second generation sequencing, specifically 454 and Illumina, we have seen that during the process of sequencing, the complementary strand is always synthesized by incorporating the nucleotides, which are either labeled with fluorophores or with some, okay, as in case of Illumina. So therefore, this is called as sequencing by synthesis. As the nucleotides are being added to the sequence, there is generation of the signal which is detected by the detector, and therefore we get to know the composition of that particular sequence. So this is sequence by synthesis. Similar to 454 and Illumina, in Ion Torrent also there is sequence by synthesis. That is, here also the nucleotides will be incorporated into the DNA strand. That means the complementary strand will be synthesized during this sequencing process. Now, why it is like that? It is like one of the reason why it is like that is because the ion torrent sequencing was invented by the same scientists, or it was developed by the same group of scientists who had developed the 454. So that is why I have written over here is that Roche 454 sequencing life sciences, it partnered with the DNA Electronics Limited and Ion Torrent Limited, leading to the development of this new sequencing technique called as the Ion Torrent sequencing technique. Therefore, you would not be surprised to find in this presentation that some of the steps during the Ion Torrent sequencing, they are almost similar to that of the what was found in case of the 454 sequencing. Okay. The ion torrent sequencing can also be called as ion semiconductor sequencing. Why it is called ion semiconductor sequencing, we will discuss in a short while. And what else was the biggest commercial hit or commercial advertisement for the ion torrent sequencing? One of the best advantage or the commercial selling point for the ion torrent sequencing was that it was first of its kind NGS sequencer, which could be put on a benchtop machine, or which could be put as at a benchtop machine inside the laboratories. So a first benchtop next generation sequencer. That means what does tell us? इसका ये मतलब है कि एक ऐसी machine which was not very humongous, which was not very huge, belonging to the second generation sequencing category, and it was a compact machine which you can put on a bench top in your own laboratory, and the chemistry is simple, and it is easy to operate. So that is why that was the uh, sellable benchmark or sellable positive point of the ion torrent sequencing, and this was lacking in the other sequencing techniques. So a positive point for that. Now, what is the basic principle of ion torrent sequencing? See, the process of ion torrent sequencing that makes it different from that of 454 sequencing is actually its principle of sequencing. मतलब कि जो प्रोसेस आयन टॉरेंट को 454 सीक्वेंसिंग से अलग बनाता है वही उसका प्रिंसिपल है. एंड व्हाट इज दैट प्रिंसिपल? दिस प्रिंसिपल ऑफ आयन टॉरेंट सीक्वेंसिंग इज बेस्ड ऑन द वेरी normal process of DNA replication. So what happens during the process of DNA replication? सबने पढ़ा है DNA replication में. What happens is that a nucleotide comes. Okay, we call it deoxynucleotide if in case of DNA. A nucleotide comes. A nucleotide has obviously purines, pyrimidines, and it has 3 phosphate groups attached to it. Okay, that is why deoxy nucleotide triphosphate. Triphosphate means there will be 3 phosphate groups attached to the nucleotide. And you can see the structure of a nucleotide. It also have a free OH group also attached to it. So what happens is that जब DNA replicate होता है, in that process, whenever a nucleotide, for example, here you can see that we are directing this nucleotides complementary to this position, such as blue always pairs with the yellow one here. So you can see that we have the yellow one here, and you can see that it is pairing with the blue one. So once the yellow nucleotide pairs with the blue one, that means the complementary nucleotides pair with each other. In that process, what happens is that the pyrophosphates are released to outside, and one phosphate is utilized to make the phosphate backbone of the DNA. Okay, so these 2 pyrophosphates, when they are released, along with them a hydrogen ion is also released. Okay, so this is the important part, that the release of hydrogen part, this proton, that is the hydrogen ion, is a very significant feature of DNA replication. So what scientists thought was that if there is a possibility that this proton or the hydrogen ion which is released during the polymerization process of or during the DNA replication process, if this proton is a chemical molecule, right? If this chemical information somehow can be converted into a digital information or a electrical signal that can be interpreted, then it will be very nice. So the whole ion torrent sequencing technology is basically based on the fact that the proton which is released in the process of DNA replication or polymerization of the DNA, this proton is this proton is utilized to make a chemical signature which is then converted into a digital signature. इसका क्या मतलब है कि ये जो प्रोटॉन है, इस प्रोटॉन को यूज किया जाता है एक सिग्नल जनरेट करने के लिए, that signal which can be interpreted. So that is the basic principle. So you have seen that कि यहाँ पर there is no chemical reaction as such, बाहर से कुछ नहीं लगा है. Okay, so what is the advantage of here? The advantage here is that कि it is different from Illumina and it is different from 454. आपने वहां पे देखा है that in that case, in case of Illumina, you can see that fluorophore are attached to the nucleotides. Okay, similarly in case of 454, a whole of chemical reaction is involved, involving luciferase, leading to the formation of the light signal or generation of a light signal. And in the light which is produced in 454 as well as Illumina, in both the cases, what do we need? We need an optical detection system to detect that light. But here, since there is no light production, यहाँ light बनी नहीं रही है, what is being formed? The protons are being formed. Okay, so the protons which are formed during the process of ion torrent sequencing, they do not need any optical detection system. But what would they be needing? They would be needing a chemical detection system, right? So that is the one difference between the Ion Torrent and other type of second generation sequencing techniques. In addition to this, we do not have to label the nucleotides or the deoxynucleotides that we are putting into the reaction. So हमें ATP, AGTC को लेबल नहीं करना है किसी फ्लोरोफोर के साथ, या किसी कोई केमिकल मॉलिक्यूल नहीं डालना है वहां पर, या कोई केमिकल रिएक्शन नहीं करवानी है उस प्रोसेस में. सो दिस इज वेरी सिंपल. इससे क्या होता है, सिंपल जो पॉलीमराइजेशन का प्रोसेस हो रहा है उस प्रोसेस में प्रोटॉन जो नेचुरली निकल रहा है हमारी बॉडी के अंदर भी निकल रहा है, और ड्यूरिंग द रिएक्शन ऑल्सो इट इज कमिंग आउट, वी आर यूटिलाइजिंग दैट प्रोटॉन. Okay, so this actually brings down the cost of the sequencing. ये sequencing को cheap बना देता है और fast बना देता है. Number of steps which are involved in the chemical conversion or in the generation of this optical signal, they are reduced, and also consequently the cost is also reduced. So that is the plus point of ion torrent sequencing. अच्छा, अब यूनिक है तो ये प्रॉपर्टी ही उसकी यूनिक है. क्या यूनिक है? द प्रॉपर्टी दैट इट इज एबल टू प्रोड्यूस protons. That means not produce protons, sorry, it is able to utilize the protons which are produced in the process of DNA polymerization or DNA replication. अब ये यूज कैसे होते हैं, ठीक है? In order to utilize these protons which are released in the process of DNA replication, there is something which is called as a semiconductor chip is involved. ठीक है? अब ये these two chips that you are seeing here and here and also here in this panel, all these are the ion torrent chips. These are called as semiconductor chips because they are called for a reason, and I'll talk about it. Okay, why they are called as semiconductor chips? As you can see that different types of chips are available: proton 1 chip, proton type 2 chip, and this is the earlier version of the sequencing chip or the semiconductor chip. इसको semiconductor chip क्यों कहते हैं? We will talk about it in a minute. Okay, so each semiconductor chip, actually what is there in the chip? If you see this chip, then each semiconductor chip, like that we had discussed in Illumina, and like that not actually Illumina, like that we had discussed in nanopores, that there were small nanopore, nanoscale holes were there. Similarly, we had also discussed in 454 that there are some wells which are present during the, which are able to present on the chip of a 454 sequencing, and in that well the bead, that is the 454 bead, can be inserted into those wells, and each well, so I hope you remember that part कि 454 sequencing में भी हमने ये किया था कि जो हमारा plate होती है, that plate has number of wells, and each of this well is capable of holding one EMPCR bead. Okay, one magnetic bead, one एक bead को वो hold कर सकता है. तो similarly मैंने कहा था आयन टॉरेंट किससे बना है? आयन टॉरेंट को भी उसी कंपनी ने बनाया है जिस कंपनी ने 454 रॉश लाइफ टेक्नोलॉजीज, ठीक है, रॉश 454 लाइफ टेक्नोलॉजी, सेम कंपनी ने बनाया 454 सीक्वेंसिंग टेक्निक को. तो ये जो सीक्वेंसिंग, दैट इज व्हाई देयर आर सिमिलैरिटी. एंड व्हाट इज द सिमिलैरिटी? सबसे पहली सिमिलैरिटी ये है कि यहाँ पर उसके फ्लो सेल की तरह हियर ऑल्सो द फ्लो सेल विच इज कॉल्ड एज द चिप हैज गॉट नंबर ऑफ स्मॉल वेल्व्स. Up the amount of well varies from the type of chip. However, this in, for example, in case of proton 1 chip, there are 165 million wells. So there are small wells, micro wells inside this semiconductor chip. And remember, like that of 454, each well is large enough to occupy only one bead. And what is that bead? That is was called as magnetic bead. If you remember, one EMPCR